I will probably like to introduce the SCAN Center. So as you know, according to me, that biggest achievement after independence of India, that we raised our life expectancy from 32 to 70. So this year, our life expectancy of India is 70. So it was 32 at 1947. So you can imagine, so that is the big things for India. But of course, there will be a lot of associated cost that probably we didn't realize because we are busy dealing with malaria, tuberculosis. So we didn't realize that our population of Alzheimer's, Parkinson's, other type of neurological disorder, aging associated disorder increased significantly. So if you look at today's scenario, India is one of the red zone in the WHO according to neurological disorder. So and these problems will increase a lot, and of course every elderly family might suffer either due to Alzheimer's and Parkinson's. So in that aspect, so at IIT kind of technological institute, we should contribute in the kind of this field, aging neurological disorder. And in this context, I will say that when we are thinking, discussing, and then as engine cannot run without fuel, Mr. Sharad Sanghi, the distinguished alumnus of our institute, he came forward and he fueled us to open this center called Sunita Sanghi Center for Aging and Neurological Diseases, and on name of his late mother, Sunita Sanghi. So thank you very much for your donation and setting up the great center, and that basically our aim how to integrate engineer, basic scientists all together and to solve some of the fundamental problem of India in terms of aging and neurology. So thank you very much. So with that, I like to show a small video. Of course, we cannot able to make Indian version of the video, but there is one video I think that can sensitize you how difficult to bear this kind of disease. So I will ask to run that video, so then you will realize how difficult to,
He's my father. I was told I need to book a follow-up appointment with Dr. Jenkins. Yeah, that's good. Okay, thank you. Bye.
Are you alright?
Yeah.
How far along are you? Well, I was just wondering how long sorry.
21 weeks on Tuesday. I'll be 21 weeks on Tuesday.
How long to go?
It's easy for you to say.
You're finding it tough. You must be excited.
I'm scared.
Of what?
Everything.
I'm sure you'll be fine.
People say that, but when I'm not. Not when.
I'm sure your husband will. You'll manage. Everybody does.
Do they?
What about your family?
It's complicated.
You don't get on.
It's just me and my dad. And he's sick. So.
It'll be okay.
diagnosis? Darlington, come on.
Anyway, I will introduce today's speaker, Dr. Umrao Monani. And first of all, I will thank you to visit us. And this is the first SCAN BSBE lecture. I will thank our Dean Research Office, Nidhi, for arranging all these things. So, Professor Umrao now Daryl C. D. Vivo Professor of Pediatric Neurology at Columbia. And he is also member Motor Neuron Center, member Columbia Transcendental Neuroscience Initiative, and also affiliated member of Zuckerman Institute at Columbia. So he did PhD Molecular Genetics, Ohio State University College of Medicine, and BS Life Science, St. Xavier's College, India, Mumbai. I was thinking whether it's Calcutta or Mumbai. Then when I was asking, is it Calcutta, he said he spent 10 years in Calcutta. So he's half Bengal. So he has lots of awards and honors, and I will not mention all of them, but a couple of them. He got 2000 Development Grand Award, Muscular Dystrophy Association of America, Young Investigator Award, American Academy of Neurology, Sanatli Aventis Innovator Award, Endowment Chair, Pediatric Neurological Sciences, and he is also currently Director, Colin Giblin Laboratory for Research on Pediatric Neurological Diseases at Columbia. So welcome, Professor Umrao.
So let me thank Samir for that kind introduction, and let me begin by saying that it's a real privilege to be presenting to members of the department and to the Sanghi Center for Research on Neurodegeneration and Aging. That was a powerful video, and it speaks to the real life clinical implications of the work that we do in the field of neurodegeneration research. I should also emphasize that I'm tremendously pleased to be back in the city where I received my undergraduate education, and from where I embarked upon a career that's been immensely satisfying. You know, when I left India 31 years ago as a young lad, Mumbai was known as Bombay and Chennai was known as Madras. And apart from a few academic institutions like TIFR and perhaps CCMB, there were not so many places that conferred PhDs in the biological sciences. Today, in contrast, there are a great many opportunities for young people interested in understanding human disease and developing treatments for these human diseases that humankind has to contend with. And it's at places like this that that journey begins. And so I'm very pleased to see so many young students enrolled in the PhD program here in the Department of Biological Sciences at IIT Bombay. So, if I had to describe the research interests of my lab in 2 sentences, I'd say that we're interested in understanding the selective vulnerability of neurons to perturbations in ubiquitously expressed proteins with functions that are generally accepted to be of a housekeeping nature. And the way that we have gone about answering that question is through the study of 2 pediatric neurological diseases. One called spinal muscular atrophy, which is a prototypical motor neuron disease and the subject of today's talk. And the other one is a disease called glucose transporter 1 deficiency syndrome, which is a disorder of the brain, and that's a more recent addition to our research portfolio. And as I said, today's talk will focus on SMA, and while the research that we do in the lab is not of a strictly clinical nature, it is ultimately driven keeping in mind the human element, that is, the individuals who are afflicted with this disease and how best to alleviate his or her suffering. So, what is spinal muscular atrophy? Some of you in this audience may have heard about this disease, but for those of you who haven't, here's a very short introduction, a very brief introduction to spinal muscular atrophy. So it's a common, frequently fatal neurodegenerative disease that is caused by a paucity of the survival of motor neuron or SMN protein, which is what you see here on this western blot. And the disease is characterized by spinal motor neuron degeneration and consequent atrophy of the proximal muscles of the limbs and the trunk muscles. So it's these trunk muscles and the proximal muscles that are affected in spinal muscular atrophy, leaving these kids quite paralyzed. And for historical reasons, SMA has been classified into 3 types, with type 1 being the most severe, type 2 being of intermediate severity, and type 3 being the most mild form of the disease. So why study SMA? Well, because it serves as an excellent paradigm to more generally study motor neuron biology and disease, as I've outlined over here. And I say that for 3 main reasons listed over here. The first has to do with its quite straightforward genetics. You see, unlike other more common neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis or ALS, which are frequently multifactorial, this is a simple, SMA is a simple monogenic disease, and so it's easier to model and arguably easier to study. A second reason I think this serves as an excellent paradigm to study motor neuron biology and disease has to do with its biochemistry. You see, the one established function of this protein that is deficient in SMA, the SMN protein, is its role in mRNA metabolism and in pre-mRNA splicing. And these, you would admit, are housekeeping functions that are important in all cells. And yet, it's these motor neurons, the spinal motor neurons, that are selectively vulnerable in this disease. And so we believe that studying the biology of SMN and SMA could shed light on the common properties of neurons that make them so susceptible, so vulnerable to death and dysfunction, stemming from perturbations in these housekeeping proteins. And last but not least, this is one of those relatively common rare diseases, if one might be permitted to describe it that way, for which we now have 3 quite promising treatments, but by no stretch of the imagination an absolute cure. And so ways in which we improve current treatments for SMA or develop new ones for this disease could serve as a template for the treatment of other more common neurodegenerative diseases, including ALS, Alzheimer's, and Parkinson's diseases. So for those of you who've never seen an SMA patient, I have a short video and a photograph of a severely affected type 1 SMA child. So these kids are never able to walk. They often present with a bell-shaped chest, and this is because of atrophy of the intercostal muscles. The diaphragm is left relatively intact in these kids. These kids exhibit very little muscle activity, extreme hypotonia, which is what you see over here, and for which this disease is also called the floppy infant syndrome. So this child is flopped over when it's being held up by the pediatrician. And any movement that you do see is restricted to the distal muscle groups, which you will see in this video in just a moment. If left untreated, these kids will succumb to disease by 2 years of age. And I think one of the most heart-rending aspects of this disease is that higher brain function is normal. So this is a disease that results in an intact mind, which is entrapped within a more or less useless body. So let me just play this video for you. And you will see that this little girl is quite paralyzed, okay? There's a little bit of movement in the extremities in the fingers and a little bit of movement in the toes, but otherwise quite paralyzed. Notice the frog leg posture. That's because of extreme weakness of the pelvic muscles. You can see this diaphragmatic breathing and this bell-shaped chest. And if you saw, there was good eye movement suggesting that this child is aware of the people around her, and that's suggestive of a normal brain function, but the child is more or less paralyzed. Now this has changed because of current treatments, but before these treatments were available, this is what a type 1 SMA child would look like, and this child would most likely succumb to the disease before 2 years of age. So what causes this disease? Well, it's caused by recessively inherited mutations in a gene known as the survival of motor neuron 1 or SMN1 gene. However, and this is a concept that I will frequently refer to over the course of the next 40 minutes or so, all SMA patients always carry at least one copy of an almost identical SMN1 paralog called the survival of motor neuron 2 gene. Now these two genes, SMN1 and SMN2, are virtually identical, save for a single nucleotide change of consequence, one that lies in exon 7 of the two genes that is depicted over here on this slide. So this particular slide explains the genetic basis of spinal muscular atrophy, and in particular it explains why SMN1 is the SMA gene and SMN2 the best known SMA modifier. So SMN1, because it has a cytosine in that exon 7 nucleotide position, produces mostly full-length SMN RNA transcripts, and these transcripts are translated into a functional and stable SMN protein, and so healthy individuals produce normal levels of the SMN protein. These healthy individuals almost invariably carry a copy of SMN1, and they may or may not carry a copy of SMN2. Now, SMA patients don't have an SMN1 gene, and they only carry one or more copies of this paralog SMN2. And SMN2, by virtue of harboring a thymine at that exon 7 nucleotide position, which is thought to create an exonic splicing silencer, produces mostly transcripts that lack exon 7 and very few transcripts that are full length. Now all of these transcripts are translated, but the ones that lack exon 7, they're translated into a protein called SMN delta 7 protein, and that protein is unstable and frequently turned over. It's rapidly degraded. And so SMN2 and by extension SMA patients produce very low levels of the functional full-length SMN protein, levels that are certainly insufficient for the survival and maintenance of the motor neurons, which eventually degenerate, resulting in an SMA phenotype similar to what you saw in that video. So as I said, much of the research in my lab stems from attempts to find better and more effective treatments for these diseases, these pediatric diseases that we study. More specifically, and in the case of SMA, the questions that have driven and that continue to drive the research in my lab are in order. First, what aspect of the neuromuscular system is affected in spinal muscular atrophy? Second, in which cell types does the SMA mutation act? Third, when does SMN have to be restored? And finally, how can the effects of reduced SMN be attenuated? Now, these questions were certainly of paramount importance when I began an independent career at Columbia about 2 decades ago, for reasons that I've outlined over here. And to a certain extent, these questions remain very relevant to date as well. At any rate, these questions will serve as a guide to the experimental results that I shall present to you over the course of the next 35 minutes or so. So I've often been stopped at this stage when I've given these kinds of talks, and I've been asked, how might one answer these questions? And one way of doing that is through the study of a good animal model, in this case, a mouse model of the disease. And since much of the rest of my talk is going to focus on what we've learned about this human disease through an analysis of these mouse models, let me very briefly walk you through what went into making them. So as I said, normal humans have an SMN1 and an SMN2 gene. Now, loss of SMN1 but retention of SMN2 results in SMA. Loss of SMN1 and SMN2 has never been reported and is embryonic lethal. Now, interestingly, humans are the only species that have an SMN2 gene. Mice don't have an SMN2 gene. And knocking out the single murine SMN gene, which is the equivalent of human SMN1, is embryonic lethal. And that doesn't do us much good when one is trying to model this disease in mice. And so we asked ourselves when we made this observation many years ago when I was a postdoc, whether human SMN2 might be able to compensate for the absence of this murine SMN gene and give us a viable, usable mouse model of the disease. And so that is exactly what we did, but we took care to introduce onto this null background a genomic copy of human SMN2. Why? Because we wanted to maintain that splicing defect, which you cannot maintain if you just introduce the human SMN2 cDNA into the mice. And so that's what we did. And when one does that, lo and behold, one does indeed rescue the embryonic lethality, and one obtains a mouse model of extremely severe spinal muscular atrophy. Now, if on that background one introduces this SMN delta 7 cDNA transgene, SMN delta 7 being the most common transcript from SMN2, then one reduces the severity of the disease somewhat and obtains what is known as the SMN delta 7 mouse model of SMA, which has really become the workhorse for many labs interested in studying this disease. And here's a video of one such mouse that I made many, many years ago. And so you can see that that's the SMA mouse, and most importantly, it exhibits the two signature features of the human disease, and that's muscle weakness and motor neuron loss. Okay? So these mice and derivatives of these mice have been very useful in allowing us to test several hypotheses about the cellular basis of spinal muscular atrophy, in order to study this human disease. And one of the first questions we addressed using these animal models was this: what aspects of the neuromuscular system is affected in spinal muscular atrophy? Very important if one wants to determine the nature of a therapeutic strategy. And all of this work, a large part of this work, goes back to a time when we didn't have treatments for spinal muscular atrophy. Okay, and this question we asked, it stemmed from the observation that in these model mice, muscle weakness seemed to precede. Thank you very much. Seemed to precede the loss of the motor neuron cell bodies in the spinal cords of these animals. Now, we found that the explanation for this apparent conundrum was quite severe and profound defects at the distal end of the motor unit, at the neuromuscular junction, similar to what you see over here. So what we found was that the neuromuscular junctions of these SMA model mice were engorged with abnormal infiltrations of the neurofilament protein, and these nerve terminals were very poorly arborized. This is what a normal nerve terminal ought to look like. And then at the post-synapse, and perhaps as a consequence of what was happening at the pre-synapse, the acetylcholine receptors, which tend to localize to the postsynaptic specialization, they tended to remain immature and plaque-like. Okay? Now, under normal circumstances, in mice, the acetylcholine receptor clusters, these end plates, begin as a plaque-like structure, similar to what you see over here. And then over a 3-week period, they ramify, they develop, they become increasingly sophisticated, and they eventually assume this nice pretzel-shaped structure, similar to what you see over here. And this transition from plaque to pretzel is arrested in these animals, so that the vast majority of these end plates continue to remain plaque-like, similar to what you see over here. Now, we found that these morphological defects at the neuromuscular junctions were reflected in electrophysiological or functional defects of the neuromuscular synapses of these mice. But perhaps, not shown over here, but described in detail in this paper that we published way back in 2008, which has been cited more than 300 times since we first published it. But I think what was most satisfying about this aspect of the work that we did was finding that these defects that we had identified at the neuromuscular junctions in these model mice were indeed reflective of what was happening in the human situation. And that's the power of these models that we study. The point is to try to understand what happens in the human disease, and we were very satisfied to find that what we were seeing over here was indeed what was happening in the human situation. So what you're looking at over here is neuromuscular synapses from an SMA patient similar to the one that you saw in that video, and you can see abnormal accumulations of neurofilament protein both in the pre-terminal axon as well as in the nerve terminal. This might best be described as a terminal sprout, which is what happens when the motor neuron that was innervating this particular end plate has degenerated. So what this particular motor neuron does is it produces a sprout that innervates this end plate. And these are normal neuromuscular junctions. And so these studies, which really showed for the first time that SMA might be more than just a motor neuron disease, that it might actually be a synaptopathy, suggested that NMJs are a key but previously unappreciated aspect of this disease, that SMN likely plays an important role in NMJ development, and that therapies designed to mitigate neurotransmission defects may be quite beneficial in the treatment of the disease. I should also point out that serum neurofilament levels are today amongst the most reliable biomarkers used to test the efficacy of currently available SMA treatments. And these neurofilament accumulations were something that we detected or we observed many years ago, as far back as 2005. This work was eventually published in 2008. So how might reduced SMN have anything to do with these neuromuscular junction defects that one sees and are so characteristic of this human disease? Well, this particular slide, which attempts to, this is a slide that depicts some of our more recent work. It attempts to establish a mechanistic link between the canonical function of SMN in mRNA metabolism, specifically in splicing, and these NMJ defects. You see, one of the genes that emerged in an RNA-seq analysis of SMA motor neurons and was found to be perturbed, more specifically to be misspliced, was agrin. And agrin, some of you might be aware, is or exists in two alternatively spliced flavors. One is called Z plus agrin, which is referred to over here as Z plus agrin, but I say Z plus out of force of habit because I've lived in the United States for so long. But so you have Z plus agrin and you have Z minus agrin. And Z plus agrin, some of you might be aware, is expressed in the motor neurons. It's secreted into the synaptic cleft, and there it acts as a very potent organizer of these acetylcholine receptors that localize to the postsynaptic specialization. And in its absence, in the absence of this Z plus agrin, what happens is these receptors, they tend to disperse into the muscle, and the postsynaptic specialization, when it's stained with alpha bungarotoxin, which is meant to bind to these acetylcholine receptors, they tend to appear somewhat dim, very similar to what we had observed in our SMA model mice. And we found that levels of the Z plus agrin isoform were indeed reduced in the motor neurons of the delta 7 SMN SMA model mice, these model mice that we were working with, which is what you see over here. And we found that levels of the Z minus agrin isoform were correspondingly increased, not shown over here, but described in detail in this paper that we published in 2017. And what is more, we showed that when you put back restore levels of the Z plus agrin isoform to the motor neurons of these SMA model mice, one was able to mitigate the severity of the disease phenotype, as evidenced by an extension of the lifespan of these animals, represented here by the trace in blue. These are Kaplan-Meier survival curves, and you'll see more of these as I move along this presentation. Also, as evidenced by an increase in the size of these acetylcholine receptor clusters. That's what you have when you restore the Z plus agrin isoform to these model mice, somewhat larger acetylcholine receptor clusters compared to animals that continue to express low levels of the Z plus agrin isoform, as evidenced by an increase in muscle fiber size and as evidenced by decreased denervation, which is very characteristically found in this disease. Again, not shown over here, but described in this paper that we published in 2017. So at least one means by which low levels of the SMN protein trigger this neuromuscular SMA phenotype is through the aberrant splicing of this NMJ organizer agrin. Okay? So a second question that was prompted in part by these neuromuscular junction defects that we found was this: in which cell types does the SMA mutation act? Very important, because it would determine the site or sites for a targeted therapy. Now, even at the time, and this is research that again goes back almost a decade, we knew that spinal muscular atrophy is characterized by motor neuron loss and skeletal muscle atrophy. And even at the time, it was clear that these NMJ defects that we had identified affected both the pre as well as the post-synaptic specialization. And so that raised the obvious question: is SMA solely a consequence of reduced SMN in the motor neurons? Is it solely a consequence of reduced SMN in the muscle, or perhaps a consequence of reduced SMN in each of these two tissues? And so to answer that question, we sought to deplete or restore this protein separately in these two tissues of wild type or SMA model mice using Krelox technology. So we began with the premise that SMA is solely a consequence of reduced SMN in the motor neurons. And to test that hypothesis, we decided to deplete the protein in just those cells. And to do that, we used an olig-2 Cre driver, and we used it in conjunction with an inducible SMN knockout allele. Okay? And olig-2 Cre drives robust Cre expression robust and selective Cre expression in the spinal motor neurons. So to deplete but not abolish SMN in the motor neurons, and the distinction is very important, because SMA is a disease caused by low levels of the SMN protein, not no levels of the SMN protein. Okay? If you completely ablate SMN in any cell type, that cell will die. That's not what SMA is. SMA is caused by low levels of the SMN protein. There are always baseline levels of SMN in SMA. And so to ensure that we had low levels but not no levels, in these experiments we made certain that these model mice that we were working with always had at least one copy of the human SMN2 gene, and that gene established baseline low levels in all tissues, particularly in tissues where our Cre of interest was active. So in these particular experiments, we began with animals that were normal, so had normal levels of SMN in all tissues, and we introduced onto this background this olig-2 Cre driver, and we ended up with animals that expressed wild type or normal levels of SMN in all tissues except their motor neurons. And we obtained what we call motor neuronal SMN mutants or olig-2-cre mutants. And we asked, what do these animals look like? So at birth, they looked indistinguishable from their control littermates, as you can see over here. But then a size and weight difference appeared at about postnatal day 15, as you can see over here, and that persisted into adulthood. Now, we tested these motor neuronal SMN mutants in a very simple assay that we have in the lab known as a writing reflex assay. It tests for muscle weakness, and you'll see a video of that in the next 10 minutes or so. What we found was that the motor neuronal SMN mutants performed quite poorly in this assay compared to their healthy control littermates represented here by the bars in gray. It's the bars in black that correspond to the motor neuronal SMN mutants. So that was true for the first week of life. But then, amazingly and quite intriguingly, these animals, these motor neuronal SMN mutants, appear to recover by about postnatal day 13, postnatal day 17, which totally surprised us at the time. I'll come back to that in just a moment. Interestingly, the animals that express ubiquitously low levels of the SMN protein, those delta 7 SMA model mice that I showed you, they continued to perform very poorly in that assay. So we also looked at adult olig-2-cre mutants, and we found that they were clearly symptomatic. Here, for instance, is one that exhibits kyphosis of the spine, which is equivalent to scoliosis of the spinal column in human SMA patients. So if you look at these patients, if they're left untreated, because of the weakness of the trunk muscles, the spinal column will assume an S shape. And so when you see these kids sitting, they aren't able to keep up the spine erect, and they assume this S-shaped spine, which is essentially scoliosis of the spinal column. And this is equivalent to what we see in the patients. We found evidence of muscle weakness in the forelimbs, in the hind limbs, and that was reflected in what we saw at the level of the myofibers. Evidence of hypertrophic fibers, similar to what you see over here, a sprinkling of atrophic fibers seen over here, and also a sprinkling of fibers with abnormal centrally located nuclei, which is what you see over here. So healthy muscle fibers tend to have nuclei that are peripherally or eccentrically located, which is what you find over here. We asked ourselves what might... Yep.
Is the organization of the gamma-sarcoglycan complex intact in these?
As far as we can tell, yes, because we stained the muscle for dystrophin, and we found that dystrophin staining was absolutely normal. Did we stain specifically for the sarcoglycans? We did not. But as far as we can tell, the expression of the sarcoglycans in the muscle is normal. Yep. So we asked what might be happening to the motor neurons in these animals, and we found evidence of spinal motor neuron loss, which is what you see over here. We found evidence of quite profound NMJ defects. Here's a fragmented acetylcholine receptor cluster. This is what it ought to look like, and this is overlaid by a nerve terminal with abnormal accumulations of this neurofilament protein. We carried out electrophysiological or functional analyses, and we found that in neonates there was evidence of neurotransmission defects shown over here, lower quantal content shown over here, and a lower evoked muscle twitch response quantified over here and shown in these two graphs. Now, interestingly, the quantal content and the twitch response appeared to recover, so that by about postnatal day 10, postnatal day 12, mutants were now no longer any different from controls. And this was consistent with the eventual equivalence between controls and mutants in that writing reflex assay. Now, to this day, we aren't absolutely certain how to explain this, but we speculate that this has something to do with a homeostatic mechanism driven by normal muscle in response to neurotransmission defects that these animals experience early on in their lives. Neurotransmission defects most likely originating in the motor neurons, because it's the motor neurons in these animals that are depleted of SMN. Muscle continues to express normal levels of the SMN protein. And this suggested to us that SMN in muscle plays an independent role in contributing to the overall SMA phenotype that you saw in that mouse and in that patient. And indeed, when we selectively depleted SMN in skeletal muscle in much the same way that we depleted it in motor neurons, we found quite profound pathology in the muscle in the form of degenerating fibers seen over here, regenerating fibers over here, and lots of little fibers with centrally located nuclei. This is the result of another experiment showing muscle damage, and what we were looking for over here was the presence of circulating IgGs that had permeated muscle fibers. It permeates damaged muscle fibers but is excluded from normal muscle fibers, which is what you see over here. And when we looked at the NMJs, we found clear evidence of NMJ defects in the form of these fragmented acetylcholine receptor clusters. Now, there are 2 points I'd like to emphasize before moving on. First of all, that this cellular pathology that we see in the so-called muscle mutants, as well as the overt phenotype that we see in these animals that include a reduced lifespan and motor defects, these are somewhat delayed in their appearance, suggesting that in SMA, defects in the motor neurons or defects that originate in the motor neurons tend to mask these other defects in peripheral organs, including defects that arise in muscle. Okay. Secondly, these defects and this sort of pathology is of real-world clinical relevance. Why do I say that? Because at least one of the treatments that is currently available for spinal muscular atrophy tends to focus on restoring SMN into the CNS. And so it restores SMN to the motor neurons, leaving the rest of the body, including muscle, essentially SMA. And that could inadvertently trigger these kinds of defects if it is not properly addressed. In fact, we have reliable evidence that some of these patients who are treated with this drug called nusinersen, these patients over time, they develop high levels of serum CK, creatine kinase. Creatine kinase is an enzyme that is expressed in the muscle and is restricted to the muscle. If the muscle is damaged, then it tends to leak out into the serum, and you have an increase in serum CK levels, exactly what these kinds of results would predict. Along these lines, we also carried out one additional set of experiments where we restored SMN selectively to the neurons. And again, this was trying to test the hypothesis that SMA is solely a consequence of reduced SMN in the neurons. Now here, what we found was that selective restoration of SMN in the neurons fails to completely rescue the SMA phenotype. In retrospect, this particular result is entirely consistent with the result that we obtained from our oligtoukri experiments, where instead of getting this full-blown SMA phenotype, we got a mild phenotype. Collectively, what these experiments suggested to us was that there are at least 2 critical cellular sites of action of the SMN protein, and that you have to therefore restore SMN to each of these 2 tissues, these tissues being skeletal muscle and the motor neurons. So these treatments that were developed thinking that SMN is only important in the motor neurons now have to be revisited. Okay, which is why we don't stop at these first generation treatments. Okay, these kinds of results then inform the development of improved treatments for this disease. Now, precisely what SMN is doing in these 2 tissues continues to be debated, but I do have some ideas about this, and I'll come to that in the last part of this talk. So, this part of our research was very focused on where is SMN really required, and therefore where does one have to restore SMN if one is developing a simple SMN repletion therapeutic strategy. But if you're developing that sort of treatment, it's also very important to know when you need to restore SMN to those tissues that need to be restored for the SMN protein. And so we also focused a large part of our work on defining the temporal requirements for the SMN protein, and we tackled this particular problem on 2 fronts. First, we asked, when does one have to turn protein expression on to rescue the disease phenotype? In other words, how long can one allow the disease to progress before a point of no return? And secondly, we asked, how long does one have to sustain wild-type levels of SMN expression to maintain a healthy state and prevent a quote-unquote relapse of the SMA phenotype? So we tackled this particular problem. So we answered these 2 questions using the following strategy. To answer the first question, we took advantage of an inducible SMN rescue allele, one that can be activated to express normal SMN in a controlled fashion. And we answered the second question by taking advantage of an inducible SMN knockout allele, so one that can be inactivated in a controlled fashion. Now let me just walk you through what this rescue allele looks like. Okay, this is a rather frightening slide to look at. It's a quite busy slide, but the important point to keep in mind is this. In its native conformation, which is what you see over here, this allele behaves like human SMN2 and produces very low levels of the SMN protein. This allele is engineered so that it can undergo Cre recombination, and this is the conformation that it assumes. And in this new configuration, it begins to behave like human SMN1 and expresses normal levels of the SMN protein. So you can see how useful this might be in turning on SMN in SMA animals, and that's exactly what we did in these sorts of experiments. So prior to effecting recombination of this particular allele, and that can be done in the presence of a tamoxifen responsive Cre-ER transgene. Okay, so prior to that, these animals express low levels of the SMN protein. One can activate the allele with tamoxifen, and one gets animals that express normal levels of the SMN protein. And one can do this at various points in time during the course of the disease. Yes? You can target it to basically any tissue. In these particular experiments, in the first instance, because we knew that SMN was ubiquitously expressed, what we did was to use a tamoxifen responsive Cre-ER transgene that was ubiquitously expressed. Okay, but yeah, you can do it to muscle, you can do it to liver, you can do, and those kinds of experiments have been done. And I'm happy to answer those questions at the end of the talk if you're interested. But over here, yes, what we were doing was we were activating SMN using this allele in all tissues. And one can do this at various points in time during the course of the disease, and then one can ask, how might that affect the disease course? Okay, and so the tamoxifen turns on the expression of the Cre-ER transgene. It expresses, when tamoxifen is administered to the animals, the Cre is expressed from the Cre-ER transgene. It is translocated into the nucleus. In the nucleus, it will affect recombination at this allele, right? And so this is what we did was we began this kind of a treatment regimen at a point in time when there was a clear and obvious disease phenotype in these animals. And that's what you see over here. So let me just play this video. And this is an example of a writing reflex assay. So this is a normal animal at P4. And you can see it's able to write itself quite quickly. This looks very much like this, but it's an SMA animal. And you can see it's paralyzed. It cannot write itself. Okay, so at P4, these animals were paralyzed. They had an obvious phenotype, and this was reflected in cellular pathology at the neuromuscular junctions. We see abnormal accumulation of the neurofilament protein. And so we took these animals, we treated them with tamoxifen to activate the allele, and then we followed them out over time, and here's what we found. A large proportion of these animals were quite substantially rescued, represented by this trace in green. So the trace in blue represents controls, trace in red represents vehicle-treated animals, and the trace in green represents animals that were administered tamoxifen. Mutants that were administered tamoxifen, and you can see that a large proportion of them are quite significantly rescued. So even at that time, and this was published a decade ago, the significance of these findings was quite obvious. It suggested to us that even symptomatic patients might benefit quite considerably from a restoration of the SMN protein, and to some extent, this has panned out to be the case. Now, at the time, we asked ourselves, okay, so you see this extension of the short lifespan, but is this really reflected in an increase in the SMN protein? Because this is how it's meant to work.
Could I ask a question?
Yeah.
So you have that pool that's clearly vulnerable, and then you have a pool that's resilient. So do you have a sense of what distinguishes that cohort from the resilient cohort?
Yeah. I think it's a combination of factors. There's always human error to begin with. So you can easily imagine that in some of these animals, the rescue allele wasn't completely turned on. It also reflects what you see in the real world, which is that there'll be, say, 10 different patients, all of whom have 2 SMN2 copies. You would expect them all to have exactly the same disease. They don't. some of them are less severely affected, some of them are more severely affected. And that raised a very interesting question that I'll come to towards the end of my talk. Okay, so it's modifiers. Some of these, in this particular instance, that's very unlikely because this is all on a more or less a pure strain background. But that's another explanation for, at least in patients, why individuals with the same SMA haplotype could have different SMA phenotypes. But the most logical and reasonable explanation over here is that some of these animals were just not fully activated for this allele. So we asked, is SMN turned on in these animals? And Western blots showed that it is indeed. It's the animals over here, the samples over here. We asked, is there a rescue of the neuromuscular junctions which are affected? And the answer to that question was once again yes. You can see that so terminal arborization at the nerve terminals is greatly improved as opposed to the vehicle-treated animals. Neurofilament accumulation is greatly reduced as compared to over here, and the postsynaptic acetylcholine receptor clusters in these animals are much larger and more sophisticated looking than those in vehicle-treated animals. So we asked ourselves what might be happening to the motor neurons in these animals, and we found that this particular treatment had restored the SMN protein to the motor neurons, both to the cytoplasm of the motor neurons, which is what you see over here, as well as to the nuclei of these cells, where SMN tends to form these bright puncta-like structures indicated by these arrows, which Gideon Dreyfus, in a moment of inspiration, decided to term gems. So he called these little dot-like structures gems. He very much wanted to rename SMN Gemin-1 because I think he found a series of proteins that he called Gemin-2 and Gemin-3 and Gemin-4, etc. The implication being that SMN1 would be renamed Gemin-1, but that never happened. Much to Gideon Dreyfus's chagrin, I think. He would never admit to that, but that's my theory. So we were able to restore SMN to both the nuclei as well as to the cytoplasm of these cells, and which is not what you see over here in the vehicle-treated animals. And we found that the numbers of these cells, which are greatly reduced in untreated animals, they're basically restored to what you see in control animals. And then when you look at these animals, what does the overt phenotype look like? Apart from being somewhat smaller than their control littermates, it's difficult to tell the rescued animals from their control littermates, which is what you see over here. And so these results, now almost a decade old, were really amongst the very first to provide proof of concept of the promise of SMN repletion as a means of treating spinal muscular atrophy. And very similar experiments which we carried out, which showed that early repletion is really the best, justified the manner in which SMA is treated today, i.e. with newborn screens and then with prompt intervention using an SMN enhancing agent. Now at the time, and as you can see over here, this was published in 2011, we didn't have these agents, but there was an effort to try to develop these agents, and our lab was very much in the forefront of that, and so we figured that that would happen. And so that raised the obvious question, once we had these agents, how long would we need to continue a treatment? Would a treatment have to be chronic, lasting the lifetime of the individual, or could it be acute, just covering a brief window in time, following which the requirements for the SMN protein might change? For instance, the requirements of the protein might fall, and so therefore the neuromuscular system, and therefore the organism as a whole, might become relatively refractory or resistant to low levels of the SMN protein. And to answer that question, we decided to deplete the protein in healthy mice at various points in time postnatally, and we did that using this very same inducible SMN knockout allele, shown over here, that we had used in our olig-2 experiments, and we used it once again with this tamoxifen responsive Cre-ER transgene. Now, in this set of experiments, we began with normal animals, treat with tamoxifen, and end up getting SMA animals. And you can make these SMA animals at various points in time. So you can make them SMA at 2 days of age, 4 days of age, 10 days of age, 30 days of age, 6 months of age, etc., etc., etc. And we asked, how might that affect the health of these animals? And the results were really quite striking. So this is how the experiment was set up, and just bear with me over here. So lots more survival curves. This is a set of survival curves of animals that were treated at postnatal day 4 in order to deplete the SMN protein. And this consists of 3 groups of animals. The first set null for SMN2, the second set hemizygous for SMN2, and the third set homozygous for SMN2. And all of them were depleted or administered tamoxifen at postnatal day 4 in order to make them essentially SMA. The animals that are null for SMN2 essentially are a control of sorts, okay? Because, as I said, complete ablation of SMN is lethal. And so we wanted to make certain that these experiments were working, and a recombination event at that inducible SMN rescue allele in animals that are null for SMN2 should completely ablate the SMN protein and ensure the death of these animals. And so that's exactly what we saw. In about 11 days or so, all of them were dead. And so this told us, yes, this experiment is working. When these animals have one copy of SMN2, then at this age, when you make them SMA, it doesn't have any effect. All of them are also dead in about 11 days. When you make them homozygous for SMN2, it does confer some benefit. These animals live out an additional 3 days or so, but again, all of them are dead by postnatal day 17. Now, this set of experiments was carried out much the same way, but here what we did was we depleted the protein during adulthood at postnatal day 50. Again, the trace in green represents those controls. All of them succumb to disease. When you make them hemizygous for SMN2, you confer a little bit of benefit. It is statistically significant. These animals live out essentially 20 days longer. But what was most surprising to us was the outcome of the experiments with the animals that were homozygous for SMN2. None of them died. None of them became symptomatic. All of them lived out to at least 6 months of age. And at that point in time, we concluded the experiments. And that raises a question which is answered by this particular Western blot that I'd like you to focus on for a second. So this is SMN protein in spinal cord tissue from these 3 groups of animals 2 weeks post-tamoxifen administration. And you can see that the levels of the protein has been depleted down to the levels that you find in the spinal cords of these delta 7 SMN SMA model mice, the animals that you saw in that video early on, the animals that are ubiquitously depleted of SMN, very severely affected. And so this told us that we were indeed depleting the SMN protein. And so this phenotype that we were seeing over here was not simply because we were getting inefficient Cre recombination. Instead, this particular difference was most likely because of the number of copies of the SMN2 gene that these animals had inherited or were endowed with in the first instance. Okay? And this is reflected in what you see over here. So these were 2 animals. Both of them were treated with tamoxifen at postnatal day 50 in order to deplete the SMN protein and then photographed 20 days later. They happened to be littermates, but one had just 1 copy of SMN2, the other had 2 copies of SMN2, and you can see that this animal is disheveled, unkempt, and rather sickly looking. But if you give this animal or a littermate 1 extra copy of SMN2, it looks relatively alert and healthy. And so there are a couple of take-home messages from this. First, that 1 copy of SMN2 is insufficient to sustain life, regardless of whether it is a neonate or an adult. The second conclusion that one, or second inference that one can make is that if you have 2 copies of SMN2, that is sufficient to sustain life, but only once adulthood is reached. And so this raised the obvious question, at what particular point in time between the neonatal stage of life and the adult stage of life do these animals become refractory to low levels of the SMN protein? And so we wanted to answer that question because, again, that is of real-world clinical relevance because it tells you perhaps there is a similar therapeutic window of opportunity in humans. It might be different, but it is very likely to occur. And that eventually did happen to be the case. So we were very interested in answering that question. But before we answered that question, we asked what might be happening to the neuromuscular junctions in these animals, which was a very clear predictor of disease severity. So this particular slide shows that the extent of cellular pathology that one sees at the neuromuscular junctions is directly reflective of the overall SMA phenotype. So lots of NMJ defects in these SMN2 null animals, fewer defects in SMN2 hemizygous animals, and few, if any, defects in SMN2 homozygous animals. And these were the sorts of defects we were looking for: fragmented acetylcholine receptor clusters and nerve terminals engorged with abnormal amounts of neurofilament protein. And so this suggested that 2 SMN2 copies protect quite significantly against NMJ defects following late postnatal SMN depletion. Incidentally, when we looked at the numbers of the motor neurons and the spinal cords of these three groups of animals, we found scant evidence of any motor neuron cell body loss. Again, suggesting that loss of the motor neuron cell bodies in SMA is a relatively late event in the disease phenotype. It is preceded by these distal defects that we see at the neuromuscular junctions. Okay? So, if depleting the SMN protein in adult SMN2 homozygous animals causes a minimal disease and cellular pathology, whereas depleting it in similarly genotyped neonates causes death, then there has to be a point in time in between when these animals become resistant to low SMN. So we asked, when does this happen? And so we depleted the protein, as you can see over here at postnatal is 4, 9, 12, 15, and 21, not shown over here, but reported in this paper that we published in 2014. And so animals that were depleted of the protein between postnatal days 4 and 12, all of them quickly succumbed. By about 2 weeks post-tamoxifen administration, all of them had succumbed. In striking contrast, just 1 of 8 animals depleted of the protein at postnatal day 15 succumbed to any form of disease, and none of the animals that was depleted of the protein at postnatal day 21, not shown over here, but reported in that article, none of those animals became symptomatic, none of them died, every one of them lived out to 6 months of age, at which point in time the experiment was concluded. And so this suggested to us that at least in the rodent, postnatal day 15 defines a threshold for a requirement for wild-type levels of the SMN protein. So depleting the protein following this point in time appears to cause minimal disease. And that suggested to us that certainly in the rodent, and perhaps who knows in the human as well, if one is able to ensure wild-type levels of the SMN protein when they are vulnerable to low levels of the SMN protein, that would ensure that a healthy state would be maintained even if SMN levels were depleted or allowed to fall later on in the disease. In other words, perhaps we might be able to envisage an acute treatment. And why is that better? Because if you have a small molecule, for instance, that is used to restore SMN, that small molecule might have adverse off-target effects, and so you want to reduce those off-target effects to the extent possible, okay? And so if you focus on an acute treatment, then you avoid long-term adverse effects. So that was the reason why we thought that this acute treatment might be preferable, okay? And incidentally, postnatal day 15 is the point, at least in rodents, at which the NMJs have completely matured. And that suggested to us that perhaps it is the state of maturity of the NMJs that dictates the organism's requirements for SMN. Now, if that is the case, then if one forces a remodeling of the NMJs, then that would unmask pathology, even if the NMJs were triggered to remodel during this apparently refractory stage in life. And so to test that hypothesis, here's the experiment that we carried out. So we took these adult animals at postnatal day 50. Half of the animals had inherited or were endowed with the Cre-ER transgene, half of them were not. The animals that were endowed with the Cre-ER transgene were engineered to express low levels of the SMN protein when subjected to tamoxifen. The animals that did not have the Cre-ER transgene would continue to express normal levels of the SMN protein, and they served as control. So we took these two groups of animals. They had 2 copies of SMN2. We administered tamoxifen, waited 2 weeks, and then we subjected them to nerve crush in order to force denervation, reinnervation, NMJ remodeling. And then 6 weeks later, we analyzed them for NMJ morphology and overt phenotypes. And here's what we saw. Whereas efficient reinnervation had occurred in animals that expressed normal levels of the SMN protein, this process essentially failed in these animals that expressed low levels of the SMN protein, which is what you see of it. Look at this. It's a fragmented acetylcholine receptor. It is not overlaid with this green, which corresponds to the nerve terminal. And this cellular pathology was accompanied by an overt phenotype as assessed by this clasping defective limb splay. So in an animal where the reinnervation has properly occurred, when you suspend them by their tails, they tend to splay their limbs like this. This is the limb where the nerve was crushed. But you can see that the animal splays out its limbs normally. If that re-innervation has not properly occurred, then this is what happens. The animals clasp their limbs. And so this suggested to us that while depleting the SMN protein during the apparently adult stage of life might not result in pathology, a triggering event like an acute injury could precipitate quite extensive damage. And so that suggested to us that SMN, or I should say expressed another way, it suggests that SMN is important both during the early postnatal stage of life when the NMJs are maturing and being refined, and also in the event of an acute injury. Now, the implications for an SMN treatment at that point in time were clear, and it led us to this kind of a model that links the postnatal requirements for the SMN protein to the state of the neuromuscular junction. So what we propose over here is that the requirements for the SMN protein are adequately met by one SMN1 copy or multiple SMN2 copies that essentially produce the same amount of SMN protein that one SMN1 does. And the requirements are greatest during this early postnatal stage of life when the NMJs are maturing and they go from this plaque-like structure to this nice pretzel-shaped structure. And these requirements then fall by about postnatal day 17 when the NMJs are fully mature and refined. And at this particular juncture, the animal enters this period where it is relatively refractory to low levels of the SMN protein. And it stays in this state until there is an acute injury or when the aging process starts kicking in, when the NMJs start turning over. Okay, they don't turn over all at once. They turn over one by one, but at some point in time, enough NMJs have turned over that you begin seeing an overt phenotype. Okay? And at that point in time, there is a surge in the requirement for the SMN protein in these motor neurons. And that surge in the requirement for the SMN protein is not adequately met by 2 copies of the SMN2 gene. And so SMN might be thought of as playing 2 roles at the neuromuscular synapse: its initial maturation and then also its continued maintenance. Okay? So what might mediate these effects? So the strategy that we adopted in order to try to find these mediators was to identify suppressors, modifiers of the SMA phenotype. And that brings me to the last part of this talk. Okay? So by now it should be clear to you that SMN2 is a major SMA modifier. Increasing SMN2 copy number decreases the severity of the SMA phenotype. But in some instances, this nice correlation between SMN2 copy number and phenotype ceases to exist. You see, in rare instances, as I was alluding to earlier, you can have so-called discordant SIBs or even a whole population of SMA patients with the exact same SMA haplotype, meaning that they all have the same SMN2 copy number but with quite distinct SMA phenotypes, suggesting that there are factors that lie outside the 5q SMA locus that might have a bearing on the overall SMA phenotype. Now, interestingly, we found that these discordant phenotypes were also a characteristic of our model mice. So, for instance, a cohort of the delta 7 SMA model mice that I have been alluding to, so a colony of these mice on a mixed genetic background and housed at the Jackson Labs that some of you might have heard of in Bar Harbor, Maine. It's a very idyllic place, beautiful surroundings, lots of trees, quite inspiring place to be in. So these mice housed at the Jackson Labs were found to be relatively mildly affected with an average survival of about 17 days. In striking contrast, the very same mice, but on a pure FVB background. FVB is a strain of mice, of white mice that we commonly use in the lab for our studies. Those exact same mice, but on that pure FVB background, and these were mice that I happened to generate on my own when I actually had time to do experiments in the lab when I first got to Columbia. These animals were much more severely affected with an average survival of just 11 days, as you can see over here. Okay? So, one way of explaining this apparent conundrum after having ruled out, by the way, these, as I said, were housed at Columbia. So one way of explaining this apparent conundrum after having ruled out obvious caveats such as this environmental influence that I was talking about, who knows, maybe the quality of the air in Bar Harbor, Maine versus in Manhattan. Manhattan is, relatively more polluted than Bar Harbor is, or who knows, maybe the quality of the care that they were being given at Columbia versus in in Bar Harbor, the folks at Bar Harbor are very gentle with their mice. The guys at Columbia, I mean, they tend to rough the mice. But, aside from jokes, that's a very real explanation. But after having ruled out that, one way of explaining this is to invoke the effect of these genetic modifiers. And so we figured if there are these modifiers, we should try to identify these. Why? Because revealing these novel disease pathways, these modifiers, would reveal these disease pathways, and that could provide genetic elements that might be targeted in future treatments. And so we went one step further, and we developed congenic SMA mice on the parental strain backgrounds FVB and C57 in black, on which the original SMA mice had been derived on. Okay, if anybody's tried this, one would know that it's a painful process, because you've got to do this over 10 generations, and it's sort of mindless work, but it needs to be done. And at the end of the day, we obtained SMA mice on a pure FEB background and SMA mice on a pure C57 black background. It's amazing that was probably about 18 months of work, and it's summarized into one slide. But so this is the important bit. What we found was that animals on the pure FEB background were much less severely affected than animals on the pure C57 black background. And that convinced us of the presence of these genetic modifiers, because in all other respects, especially in terms of SMN2 copy number, they were identical. So what might explain this? So we decided to identify precisely what might explain this. And the way that one goes about doing that, again, it's a long drawn out process, but this is a schematic. What one does is one takes animals on the mild background and animals on the severe background, SMA carriers, one crosses them, one generates SMA mice, and one asks, how do they segregate into animals with different phenotypes? And this is what we obtain. So most of the animals presented with what I would call a typical severe SMA phenotype, with an average survival of about 2 weeks. But a small proportion, about 5% of these animals, not only did they attain the maximum age of 27 days attained by these guys, but then went on to survive to an average of like 120 days, like a more than 10-fold increase in survival. And so what we did was we used these two cohorts of mice to try to identify precisely what might be responsible for this. So we began with a classic linkage analysis. This is where the genetics background comes in, and here's what we saw. There was a region on chromosome 9 that derived from the C57 black strain of mice that was closely linked to this mild or modified SMA phenotype. And so what did we do? We took this region of chromosome 9 from the C57 black strain, we put that and that alone onto an otherwise FVB strain background. We made carriers, SMA carriers, and then we crossed them in order to generate SMA mutants, and here's what we found. So these congenic chromosome 9 SMA mice were found to exhibit pretty much the same sort of modified SMA phenotype, mild phenotype, that we had observed on the mixed genetic background, as evidenced by a big extension in lifespan. So in these experiments, we found that these animals some of them were able to go to 450 days, and eventually we found that some of them went out to 600 days, as compared to this. The severely affected animals only survived to about 10 days, 12 days, okay? As evidenced by improved motor performance, you can see in this writing reflex assay, the modified mutants now perform just as well as their healthy littermate controls, and as evidenced by a restoration of the motor neurons in the spinal cords of these animals. And so this told us that, yes, indeed, this region of chromosome 9 most likely contains the modifier or modifiers that we are interested in. In other words, it is a region of interest. And here's one such modified animal. Now, I'd be the first one to admit that this animal is not entirely normal. This one, for instance, has a necrotic tail. But by the same token, this animal looks nothing at all like the animal that you saw in that video. Now, there's a standard way of narrowing down such regions of interest, because this was still a fairly large chunk of chromosome 9, but there's a standard way of narrowing down such regions of interest and identifying the factor or factors that are responsible for this, okay? Again, it can be a long and arduous process, and in our case it was, but that was mostly because we were sort of naive to these kinds of experiments. We hadn't done these kinds of experiments. But based on the sequencing of a critical 4 megabase pair region of interest, and also based on some key recombinants, we were able to identify a synaptic chaperone that was invariably mutated or altered in our modified mutants, but generally wild type in the typically affected mutants. And so what did we do? We used CRISPR-Cas9 to alter the wild type version of the synaptic chaperone on the otherwise severe background. We created carriers, we used the carriers to generate mutants, and then we waited with bated breath to see what the mutants would turn out to be like. And the data on this particular slide provides unequivocal evidence that the synaptic chaperone is indeed the genetic suppressor that we were after. Once again, as evidenced by this big extension in lifespan, increased body weight. So typically severe SMA mutants rarely get to beyond about 2.5 grams in weight. It's the trace in red, and that trace in green represents animals that are homozygous for the modifier, and as evidenced by improved writing ability. I should point out that even one copy of the modifier does significantly modify the SMA phenotype. Most obvious over here. It's less obvious in the survival curve, then that's because of the scale of the x-axis. But you really require 2 copies of the modifier to have the most potent effect. The other thing that I would emphasize is nothing at all, in my experience, has modified the severe SMA phenotype to the extent that this modifier has. It is sufficient on its own to accomplish this sort of modification. There are other modifiers that have been identified, but the only way they work is if you begin by restoring to the animals a little bit of SMN in some means. Then those modifiers work. In this particular case, the modifier works entirely on its own. Okay, so we've begun to look at the mechanisms of action of this modifier, and I can certainly answer this during the question and answer session, but I think I am running out of time, and so I'll try to wrap things up over here and say that, you know, over a
question. The function of this chaperone is in the muscle which this axon is trying to target, or because in the beginning you said that we need to put in the muscle, right?
So the protein is required in both tissues. That is a given. I am absolutely convinced of that, okay, from multiple observations that we've made. So that raises the question, where does this modifier work? Now, as I said, I'd be the first one to admit that the animals that express this modifier are not completely rescued. They are very significantly rescued. I think that this modifier primarily functions in the motor neuron. And I think it works in the motor neuron because of several lines of evidence that suggest that this modifier is extremely important in the establishment and regeneration of snare complexes. Snare complexes, some of you might be aware, are these complexes that are within synapses that are very important for sustained neurotransmission. If these snare complexes are disrupted, neurotransmission is disrupted. Neurotransmission is disrupted in SMA. But if you look at neurotransmission in these mutants, it is not only restored, it is actually potentiated. So I think this modifier works primarily in the motor neuron. Does it have a function in other cells? It probably does, but I think it works primarily in the motor neuron. Over this 2-decade period of life, we've used these model mice to uncover this novel aspect of the disease phenotype, these NMJ defects suggesting a new avenue for the treatment of the disease. We have used these model mice to define at least 2 critical cellular sites of action of the SMN protein, motor neurons and muscle, suggesting that you've got to restore SMN to both of these 2 tissues if you're envisaging a SMN repletion treatment. We've used these mice to demonstrate that treatment may be initiated even after symptom onset, and even then the treatment could be effective. And we've simultaneously shown that once the organism attains adulthood, it becomes relatively refractory to low levels of the SMN protein, with the caveat that if the NMJ undergoes acute injury, then there's going to be this surge in requirement for the SMN protein, and that's when you need to ensure that the motor neuron is provided with SMN. So what you can do is you can probably design the treatment to be acute, but then provide it on an as needed basis. Okay, and we have used these model mice to identify this disease modifier, which promises not only to reveal the basic mechanisms about SMA, but also could serve as a new therapeutic target for the treatment of this disease. Now, I would be completely remiss if I did not acknowledge the people who did all this work. I mean, this is really work that was carried out over 2 years, and so let me end with this particular slide. These are contributors past and present. That is a photograph of us following the award of our first big NIH grant. You can see all of us have a delightful look on our faces, and that's me looking much younger and quite happy. And that's a more recent photograph of us and the current complement of the lab. I've lost a lot of my hair, and what remains has gone quite gray, but we're nevertheless a quite happy lot. And if you want to be featured on such a slide, please don't hesitate to contact me at um2105@columbia.edu. We're always looking for bright young postdocs and motivated postdocs, which I talked about this afternoon. Thank you for your attention. Happy to take questions, and thank you for inviting me.
During formation of the neuromuscular junctions, the neuron already has SMN protein. So does the muscle. And before I heard your talk, my assumption was that the neuron is the one establishing contact with the muscle to form the neuromuscular junction. But it now looks like the SMN1 from the muscle itself is also required to consolidate this neuromuscular junction at the site where the neuron makes this connection. So what is coming from the muscle that consolidates or matures, that allows the neuromuscular junction to mature?
I mean, I think the simplistic answer to that is, if you have muscle that is deprived of the SMN protein, it is an unhealthy muscle. If it is an unhealthy muscle, the crosstalk between the neuron and the muscle is affected. If it is affected, there are various factors that the muscle secretes in order to talk to the motor neuron. There are various factors that the motor neuron secretes, and all this happens in the synaptic cleft. And you can easily imagine that if the muscle is deprived of SMN, some of these factors are probably not secreted. Some of these factors that are required for the establishment of the healthy neuromuscular synapses, my guess is that some of these factors, what we'd be very interested in determining is, how the loss of SMN in the muscle affects some of these factors that are normally secreted by the muscle to ensure that the motor neuron remains happy.
Yeah. My second question was, has anyone tried, let's say, stimulating muscle contraction or electrical activity in the muscle to, restore NMJ maturation in SMA models or patients?
So that's a good question.
So is NMJ maturation muscle activity dependent? And if so, could you restore muscle activity to force maturation of NMJ?
So what we find is the muscle mutants that I talked about, that's a good model system in order to answer that question. What we find is that the neuromuscular junctions are indeed established properly during early postnatal development. The neuromuscular junction is not maintained in those mutants. I think the amount of SMN in the muscle of those mutants is sufficient to establish the neuromuscular junction. It is not sufficient to maintain the establishment of the neuromuscular junction. So my guess is in the first 3 or 4 weeks of life, the muscle is able to secrete those factors, or perhaps the requirement by the motor neuron of those factors is not that high. But then over time, the motor neuron says, okay, I'm talking to somebody over here, but somebody is not answering. And that somebody who's not answering is the muscle. And at that point in time, you begin to see defects both in the presynapse as well as the postsynapse.
So I had a question with reference to oculomotor, facial nerves, etc. I mean, this is all spinal motor neurons. What's happening to neuromuscular junctions of the face, the eye, etc.? Why are those spared? And why do you have no requirement of SMN in that population of cranial nerves?
Again, I want to make the distinction between no SMN and low SMN. So, yes, so those motor neurons are relatively spared, okay? But over time, and there is a cohort of, I would say, unfortunate type 1 SMA patients. If you let these individuals go out for sufficiently long, all of their motor neurons, including the ocular motor neurons, degenerate. That still leaves the question, why do they survive for such a long time when all these other motor neurons degenerate? And I think that's one of the burning questions, not just in the field of SMA, but also in the field of ALS. So for instance, in ALS, it's the alpha motor neurons that are most severely affected. The gamma motor neurons are not. That is a question that remains to be answered, and the honest answer is I don't know.
Fascinating.
But what is also quite, so in this context, what I will say is, so as I also said, the brain is left relatively intact in this disease, but the brain also consists of lots and lots of synapses. But why is brain function more or less intact when you have these neuromuscular synapses that are so profoundly affected? Again, one simplistic answer is that the brain synapses are relatively small compared to the huge neuromuscular synapses. But what's also very interesting, and I don't have an explanation for this, when you look at defective snake complex assembly, it's the neuromuscular synapses that are affected. It's not the brain synapses. And there are experiments that have been done by Tom Sudoff and various other people which have looked at the effect of depleting various synaptic proteins on the neuromuscular synapse on the one hand and the brain synapse on the other. And actually, the way that these two synapses respond can be quite different, even though the essential machinery is quite highly conserved in brain and neuromuscular synapses. I think that's going to be quite important to and interesting to tease apart.
Hi. Really enjoyed your talk. I just have a couple of questions actually. The first one might come as naive. So
no question is
naive.
naive.
Talk about crosstalk between the motor neuron and the skeletal muscle, and I read about the study where when the motor neuron is going in and arborizing or making contact with the skeletal muscle, it releases acetylcholine, and one of the main players is CAT, which is present on the skeletal muscle. And there was this study done in 2005.
The protein you said was JA?
CHAT, which is
CHAT. Oh, okay, yeah.
And one of the studies that they did in 2005 was they knocked out actin and CHAT, like individually. There were problems in arborization, but when they knocked out both of them, arborization was just fine. So I was wondering here as well, like SMN interacts through actin. So if you could also knock out CHAT, for example, in skeletal muscle, if that solve the problem.
My gut feeling is that, I mean, if you see any sort of compensation, my guess is that it's going to be very temporary. In order for you to, have a healthy neuromuscular synapse, I can't think how, adding insult to injury would make the situation better. I think what those kinds of experiments that you're referring to suggest is that, in the initial establishment of the synapse, if you prevent the one from reacting to an absence of the other on the other side, you may establish it in what looks like a normal fashion, but I don't think it'll be maintained. I don't think that was, so the experiments that you're referring to, I don't think they were done in the context of SMA. I think it was probably done in the context of just looking at the normal function of the neuromuscular synapse. That's right. Do you know how long they looked at these? Was it done in vivo? Was it done in vitro?
So these were in vivo experiments, which is not visual limitation, but they also looked at it physiologically and function was intact. So I was just wondering in this case as well, if it will solve the problem.
I mean, I think, so it hasn't been done in the context of SMA. I think it would be an interesting experiment to do, and if there are people who are interested in collaborating, we can certainly make these models available to them. I think that, in the first instance, it might be quicker and maybe simpler to do it in a cell culture model. And these are the sorts of experiments that Lynn Landmesser did. I mean, she collaborated with us many years ago. So she did it in the context of other proteins. And so, she made the motor neuron deficient in these proteins on the one hand, the muscle deficient in the protein, and then she would look for establishment of these contacts and then the maintenance of these contacts. In vitro, the neuromuscular synapses never really form the way that they would in vivo, because it's not just the motor neurons and the muscle. You need terminal Schwann cells and pericytes and all sorts of other cells. But you still, I think you get a sense of, the requirement of the protein in the presynaptic specialization and the postsynaptic specialization, and maybe coming from other cell types as well. So, one could probably try to address that in the first instance in these cell culture models, and then move on to these, perhaps in the mice or in zebrafish or in C. elegans or what have you.
Right. I just have another question. So you did talk about in human patients, even though you have the same type of genetics, you present with very different extent of severity. And one of them, as you said, might be due to genetic modifier. Specifically, I was wondering if you were also looking at epigenetics or methylation, chromatin modification,
Right.
any kind of.
I mean, I think that's a great question. And I think that before one starts getting too complicated and saying, well, it's this modifier and that modifier, you just ask them some simple question. Okay, yes, patient A has 2 SMN2 copies, patient B has 2 SMN2 copies. They have different SMA phenotypes. Let's just answer the simple question. Are the 2 SMN2 copies in patient A behaving exactly and expressing exactly the way that the 2 copies of SMN2 are expressing in patient B? They may not be expressing the same way. It may be that patient B is less severe just because he has these 2 SMN2 copies that are expressing higher levels of the SMN protein. So that's absolutely a reason why you could have these discordant phenotypes. But having said that, there are also individuals where, as far as we can tell, this is especially in SIBs, okay? And in some cases, yeah, it's mostly in SIBs. So in these cases, the SMN2 copies are unlikely to be different, okay? And as far as we can tell, the amounts of SMN protein that are being expressed, at least in fibroblasts from these patients, are not different. In those instances, it's probably something that is acting in an SMN independent fashion. If there is a difference in SMN levels, it could originate in the SMN2 copies themselves, or it could be, as you said, something that is affecting, something that is outside the SMA locus that is affecting the expression of the SMN2 copies. Because, so there's a particular variant in the SMN2 gene, it's an intronic variant, and it just results in higher levels of the full-length SMN transcript. That's also an explanation for these discordant phenotypes. But, from a basic biology standpoint, we are interested in getting beyond just SMN levels as a modifier of the SMA phenotype and asking, so what I can tell you, for instance, this modifier that we've identified, it has multiple mechanisms of action. Initially, we thought that it doesn't seem to have any effect on SMN. So we looked at these animals at their SMN levels, and the SMN levels were again way down. And so we thought this has to be acting in some other fashion. At the time that we made the original observations, we couldn't really segregate the cohorts of these mutants into animals that would become modified and animals that would remain severe. We had to do all of these experiments in age-matched controls. We were never able to really tell at P9 whether one cohort would become mild and one cohort. After we found this modifier, then we were able to simply genotype it. Then we looked at the levels of SMN, and we found that the modifier does indeed result in slightly higher levels of SMN in the modified mutants. But if in SMA, in the typically affected animals, you have about 10% of wild-type levels of the SMN protein being expressed, in the modified animals, you have maybe, I'd say, 12% or 14% of SMN. And just intuitively, I can't imagine that that tiny increase in SMN results in this very profound mitigation of the SMA phenotype. And then when we started looking at other functions that have been attributed to this, and especially looking at snare complex assembly, etc. And when we did some experiments where we started overexpressing the chaperone in a cell culture model system, where you look at the effect of it on snare complex assembly, there was a clear and obvious increase in snare complex assembly. So we can't but help think that it acts at different levels, one of which is in potentiating this. So I have a suspicion that this also happens in human patients. What's been challenging and I think a criticism of these studies is that we haven't been able to find this in human patients as yet. This is not to say that it doesn't occur, but coming by these patients and obtaining patient material is tough. But I think if we do it in a concerted way, I wouldn't be surprised if we find this. Yes.
So just a follow-up question on what we asked. Timoxifene is known to be estrogen receptor modulator. How it is working in your case? That was not clear.
Yeah, so the Cre-ER transgene, this has to do with the biology of this Cre-ER transgene. The Cre-ER transgene is a modified, it contains the estrogen receptor. The tamoxifen binds to the estrogen receptor, changes the conformation of the estrogen receptor. From the Cre-ER transgene, this estrogen receptor is excluded from the nucleus. It is in the cytoplasm. When the tamoxifen binds to it, it changes the conformation of the estrogen receptor. The estrogen receptor is translocated into the nucleus. Over there, it effects recombination. So the tamoxifen merely acts to change the conformation of the estrogen receptor so that it can go inside and effect recombination of these alleles, whichever allele you're interested in.
And this is precisely like, it is known in breast cancer, like treating breast cancer. Is it specific in MSA that you are?
In SMA? Yeah, so I mean, this is the Cre-ER transgene is merely a tool that allows you to change the expression, effect the recombination at these inducible alleles and to knock them out or to express them in a controlled way. It's merely a tool. It's that kind of estrogen receptor would never, I mean, to my knowledge, it's not of physiological relevance other than to cause these, I don't know, biological tricks, if you may. That's it.
What's the difference in this modifier, the chaperone, in the severely affected mice and the mice which survive for long? What's the actual?
So it is it's a missense mutation in the substrate binding domain of the chaperone, and that is of physiological consequence because what it does is it changes the affinity of the chaperone for its co-chaperones that are extremely important in the synapse to ensure the formation of the snare complex. So it affects the binding of the chaperone to these co-chaperones, the co-chaperones being a protein called SGT and CSP. That's one effect. And it also seems to have an effect on the affinity with which the chaperone binds SMN itself. So it enhances the interaction of the synaptic chaperone with the co-chaperones, and it decreases the affinity of the chaperone for SMN.
It's like loss of function and gain of function.
Yeah, I think that's, yeah.
I was wondering if there have been studies to compare, let's say, spliceosome profiles between SMN1 mutants and wild type cells, because the protein is ubiquitously expressed, right? But in an SMN1 deleted condition, have people compared spliceosome or splicing defects in neurons versus other cell types?
That was a very early effort, because the function that was attributed to SMN in the first instance was in the biogenesis of these snRNP, small nuclear ribonuclear protein particles, and in pre-mRNA splicing. So the next obvious question, okay, so if that's the case, then let's look to identify what might be the critical splicing defects in SMA compared to but there are a multitude of changes that occur, and it changes depending on whether you're looking at severe SMA, intermediate SMA, or mild SMA. We focused on agrin, and clearly from our results, agrin is not the whole story, because if you restore agrin, it only has a mild effect. There are clearly other splicing defects that occur. What we found is that in these modified animals, you have a restoration of neurotransmission, and yet some of these splicing defects that have been identified in the past as being immutably linked to SMA, those splicing defects remain. And so what that suggests is that SMA is not immutably linked to these splicing defects. These splicing defects can remain, and neurotransmission is nevertheless restored. So I think that's a difficult experiment to do, and if I was to do those kinds of experiments, what I would try to do is to focus on the milder forms of SMA and ask whether in those forms of SMA where there is still a neuromuscular SMA phenotype, what are the splicing defects in those forms of SMA? Because there, all the other peripheral defects and all the other defects that you find in severe SMA, including, for instance, defects in these oculomotor neurons and skin and liver and kidney and what have you, all those are corrected. It's just the neuromuscular defects that remained, that persist. So in that disease, let's look at the spliceosome. And what's interesting is that this modifier is a member of the spliceosome. Yeah, it's quite interesting. I mean, nobody knew of this and nobody sort of focused on it, but I went back into the literature, and when you do pull-down studies, it appears in the spliceosome. And there's some very old work, just like Vidita was talking about, serotonin being reported in plants, and people who are working on model mice, it's like, we don't talk to the guys who work on plants, and yet it's been out there. So in that fashion, it had been reported a long time ago that SMN interacts with this particular chaperone. And the thing is, I asked a very well-respected scientist who found this, and he said, no, that's an artifact. And I said to him, no, that's not an artifact. That's real, and it is of physiological relevance. But he was looking at it from a purely biochemical point of view, and I think that just sort of emphasizes the importance of, I know it sounds terribly cliched, break down the silos.
Any other questions? So okay, so then let's thank Umrao for a fascinating talk and about SMA and the neurological disorder, and Great. Thank you very much.
Thank you.
Come, please.
Oh my goodness. This is.
They are taking a photo maybe. Yeah.
All right. Mr. Stand over here.
I'll take my mask off at least so that
This is a great privilege, and if I didn't have the brown skin that I do, you'd probably see me blushing. I think it's absolutely wonderful that Mr. Sanghi has endowed the center to do research on neurodegeneration, because as was emphasized, I think that, and as I was sort of referring to early on with the discussion that I had with students, I think this is going to become a greater and greater problem. We've been very focused in the West on neurodegeneration, but this is inescapable. I think that these communicable diseases that kept lifespans in developing countries short and sort of masked the appearance of these neurodegenerative diseases. I think many of these communicable diseases, and it may sound ironical in the state in which we are today, where we are having to contend with COVID, but nevertheless, I think that we have these vaccines, and I think that many of these communicable diseases will be addressed, and so we are then going to see more and more individuals suffer from these neurodegenerative diseases. And I think it's going to become an enormous societal burden, and I think this is really the time. I mean, we should have started working on this decades ago, but sooner rather than later, and I think it's a tribute, an absolute tribute to people like yourself, that you've recognized this and endowed the center, and I think only good things will come of it.
Thank you. Thank you. Thank you.